Hi, I'm Dr. Kirthana from India. Title of the paper is Scientific Analysis of Electrophysiological Effects and Anxiety Reduction with 3 different Indian melodic scales. Music, being an integral part of our lives, is predominantly used as an entertainment modality across the world. It has an effect on the heart and the various parts of the brain as shown. However, this scientific evidence comes from the study of different types of music, especially so the Western classical music composed by the various composers Mozart, Beethoven, Bach, Vivaldi, etc. Scientific evidence of Indian music is quite meager. Ancient music literature, that is, the Gandharva Veda, lists a number of melodic scales of Indian music which are called as ragas, said to have health benefits. However, the scientific evidence in the form of study of electrophysiological effects or anxiety reduction or any other form of stress is very meager. Like Western classical music, which has do re mi fa sol la ti, we have sa re ga ma pa dha ni with their subtones as shown in the picture. The various combinations of these notes results in formation of melodic scales. In Indian music, they are called as ragas. Objectives of the present study was to evaluate the acute effect of passive listening to Indian melodic scales on various electrophysiological parameters, which included heart rate variability, electroencephalography, event-related potentials, blood pressure, state trait anxiety inventory anxiety study, as well as salivary stress markers evaluation. For this, we conducted a randomized triple-blind control trial on 4 groups, of which ABC was the music intervention group and the D was control group. As shown here, over a duration of 30 minutes, we recorded the various electrophysiological parameters. We did lead to ECG, a 19-channel EEG recording with appropriate event marking. We also recorded the blood pressure and collected saliva using salivary oral swabs and then measured the cortisol and the amylase levels. As shown over here, we recorded all these parameters over a duration of 30 minutes, of which the first 10 minutes and the last 10 minutes was without any intervention and the mid 10 minutes had the intervention. These are the 3 Indian melodic scales that were used. Group A received Raga Miyaki Todi, which is a heptatonic scale, whereas Group B received a pentatonic scale named as Raga Mal Counts, and Group C received a hexatonic scale named as Raga Puriya. Results. A significant reduction in state anxiety was observed with all the 3 music interventions, in that Raga Puriya had the maximal significant reduction in state anxiety after the intervention. Insignificant changes were observed in blood pressure and salivary amylase levels reduced in all the 4 groups. Coming to heart rate variability, an arousal effect was observed using Raga Miyaki Todi and Raga Puriya during intervention. However, both these 2 ragas caused significant relaxation after the intervention. In comparison, Raga Mal counts and the control group had a sustained increase in parasympathetic parameters over a duration of 30 minutes. As shown over here, heart rate reduced in all the 4 groups. However, in the control group, after the intervention, heart rate change was not much. Coming to this graph, which shows one of the robust parasympathetic indicator RMSST, root mean square of the RR intervals. What is shown over here is that the A and the C group, that is Raga Miyaki Todi and Raga Puriya, caused an arousal effect via reduction of RMSST during intervention and maximal relaxation, which was seen after the intervention with rise in RMSST. Coming to the EEG changes, we see that group A, B, C and D have been shown as 4 different rows in these topographic maps, and the conditions before, during and after intervention has been shown as 3 separate columns. This picture shows the delta power spectral changes, which was significantly changing only in group C, that too after intervention. Whereas in theta band, we observed that group B and C both had significant changes in the various electrodes. The red dots indicate significant changes. Coming to the alpha and the beta 1 power spectral changes, we observed that both group A and B showed significant reduction in alpha and beta 1, especially during intervention. P300, we observed that the P300 amplitude reduced and the latency increased with music intervention. Particularly so, the amplitude reduction continued after intervention in group B, whereas the latency increase continued even after intervention in group C, that is with Raga Puriya. This could be due to the lesser concentration on the task that was provided for ERP recordings or more of internal attention among the subjects while listening to music. To summarize, music intervention overall reduced the anxiety levels significantly. Passive listening to music is thus beneficial for anxious patients. The different electrophysiological changes that were observed with each melodic scale and the plausible mechanism that can be proposed in this study is that in a similar way like the Western melodic scale, due to the presence of major and minor tones and the different combinations of these major and minor tones in each of these melodic scales, the electrophysiological changes also varied with each melodic scale. To note, major tones are said to produce a pleasant experience, whereas minor tones are said to produce a sad experience. To conclude, Indian music produced a relaxation response similar to Western classical music. Of the various Hindustani ragas that we used, Raga Puriya was found to be maximally beneficial with anxiety reduction, parasympathetic response after intervention, and theta power increase during intervention. However, Raga Miyaki Todi and Mal counts also caused relaxation, just that the timing was different. Thus, further studies are highly recommended in order to build a scientific foundation for the use of Indian music in medicine. I thank the funding agencies ICMR and RGHS Government of Karnataka and Government of India for funding this project. I also thank the organizers of Future Physiology UK 2021 conference. Thank you all.